Hey everybody, so we are going to continue talking about DNA and we're going to focus on some of the details of replication today. So replication has to start at a specific spot or spots depending on what kind of DNA you're looking at. So we call those spots the origins of replication, and they are specific sites on DNA where replication starts, and they're recognized by a specific DNA base sequence. Now, I said it depends on what kind of DNA you're looking at. So if you're looking at a prokaryotic cell, those types of cells, prokaryotes, are going to have circular DNA. And so because of that, there is one origin site. So replication is going to start in one spot and run in both directions, away from the origin site until they meet up again on the other side of that circular DNA molecule. So here's an example of this. So you can see we've got our origin of replication right here. The dark blue is going to be the parental or template strand of DNA, and then the light blue is going to be that new daughter strand. And so you can see replication is starting here, moving in both directions, so it's moving this way, it's moving that way. We call this area where replication is occurring that replication bubble. You can kind of looks like it's bubbling up. And where the parental DNA is separating, we call that the replication fork. So the replication fork is going to move in both directions, so this one's going to continue moving this way, over here, this one's going to keep opening up that way, and then eventually you are going to end up with completely copied DNA, and so that's going to make two DNA daughter molecules. In eukaryotic cells, it's a little bit different because we don't have that circular DNA, and so you have many origin sites. And the replication bubbles are going to fuse to form new DNA strands. So this kind of makes more sense if you can see it. So here is a parental strand of DNA. So you can see it's a line this time instead of being circular. You have multiple origins of replication. You're going to have a replication fork on either side, and then that's going to form your bubble. Replication is going to continue at all of these different origins until eventually you can see here those bubbles are expanding, they're getting closer together, and eventually these new DNA molecules will fuse together and you'll have the two daughter DNA molecules. So it looks a little bit different than the prokaryotic, and you're having lots of spots where replication is happening instead of just starting in one spot and then spreading towards each other. So DNA elongation now is the next sort of general step, and we're going to look at specifically the enzymes that help DNA elongate, and we're going to look at this process and how this happens. DNA elongation uses enzymes called DNA polymerases. Probably the big one we're going to talk about the most is DNA polymerase 3. And so this is going to add a DNA triphosphate monomer to the growing replication strand or the new strand of DNA that's being built. And it's matching those A's and T's and C's and G's together. Now, because you're building a molecule, you're going to need energy for this to happen. And so the energy is actually going to come from these triphosphate monomers. They're going to lose 2 phosphates as each monomer is added. What that means is you have your triphosphate nucleoside monomer right here. Okay. As this is added, so in the presence of DNA polymerase, 2 of these phosphate groups are going to leave, and so what you're left with is 1 phosphate that's going to bind to the sugar of the monomer just above it. So we're adding thymine here, so we have our thymine, our sugar, our 3 phosphate groups. When DNA polymerase is there, it's going to remove 2 of those phosphate groups, and that 1 remaining is going to attach to the sugar from the nucleotide just above it. One of the things, I'm going to go back really quick, I want you guys to notice is that it says 5' and then 3', and then up here it says 3' and then 5'. So that is, we talked a little bit in class about anti-parallel. That is showing that these strands of DNA are anti-parallel. And so what that means is it's sort of like cars on a two-way street, right? So they're moving parallel to each other, but they're moving in opposite directions. DNA does the same thing. So if one strand is going 5' to 3', then the other strand, it will be opposite, so 5' to 3'. So DNA can only elongate in that 5' to 3' direction. So if we're looking at that, we've got our 5' end to 3 prime. So you can only build going in that direction. Why do we call it 5 prime and 3 prime? It has to do with the carbons. So here we're kind of zoomed in on one of these sugars. And so this is our 5th carbon. This is our 3rd carbon. When DNA is all built and these nucleotides are attached to each other, you're always going to have the 5 prime and then the 3 prime, and it's always going to be aligned that way. Just for sake of remembering this, I always think of the 5 prime and phosphate. So you've got the same kind of sound there, 5 prime and phosphate. So when you line these up, where you have your kind of hanging phosphate group, that's your 5 prime end, and then the end where you don't have a phosphate group, that is your 3 prime end. So you can see here this idea of anti-parallel. So it's starting with the phosphate, and it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. Okay. In the anti-parallel strand, if you're starting at the top, it's going to start with the sugar first. So you've got now your sugar, phosphate, sugar, phosphate, sugar, phosphate. So the more you guys practice kind of modeling this, I think the easier it gets to remember. But again, I always think 5 prime has your hanging phosphate. Okay, so now we want to talk a little bit more about the 2 different strands and how they're copied. So you're going to have a leading strand, and this is going to be the continuous replication toward the replication fork, and this is going to be moving in that 5 prime to 3 prime direction. Now keep in mind that DNA polymerase III, which is the enzyme responsible for adding these nucleotides, can't just start this DNA synthesis. Before DNA synthesis can start, before it can start adding these nucleotides, you need to have a primer. Sort of like painting your house, right? You have to put a primer down if you want the paint to stick. You have to put a primer down here if you want to add the nucleotides to the DNA. So the primer is going to be made of RNA. It's 10 nucleotides long. It's added to DNA by an enzyme called primase. And then once that primer is there, then the DNA is added to the RNA primer. And this needs to happen for every single DNA elongation site. This isn't just happening once. This is happening over and over and over again. Every time you have a new replication, or I'm sorry, a new origin of replication, you have to prime it first, and then you can start adding the DNA. So the important parts that you're looking at right here, this is your DNA polymerase. Okay, this is your 5 prime to 3 prime going this way. And so if this one starts with 5 prime and goes to 3 prime, then that means that this complementary strand that's being built is going to go from 5 prime to 3 prime. So this DNA polymerase is just going to keep moving towards the replication fork and adding DNA bases. So this is your leading strand. You can see you've got your RNA primer right here, and then after this primer, that is where the DNA polymerase starts adding the DNA nucleotides. And then down here, again, we still have our primer, and you can just see now that the DNA polymerase III has just continued traveling towards the replication fork, so there is a much longer strand of bases that have been added. So now we want to talk about the lagging strand, and this is going to be the discontinuous synthesis, and it's going to move away from the replication fork. So you're replicating here short segments as more of the template opens up. We call these short segments Okazaki fragments. They're like 100 to 200 bases. Again, they're added onto the lagging strand. All of these fragments must be primed the exact same way that we talked about, and then later on that RNA primer is going to be removed once it's ready to kind of be done with this replication. So we're moving in the 5 prime to 3 prime again. So here's our primer. Then we have DNA polymerase III adding the bases. So here we have one finished Okazaki fragment, and we'll have it kind of bumping up into another primer from a fragment that was already made. Now we need to reprime right here, and we will make this strand of DNA. Once that's complete, then we're going to come in and we're going to replace the RNA primer with DNA bases, and then we're going to seal it all up so that this is all one continuous strand of new DNA. So the enzymes that are involved in this are DNA polymerase I, and DNA polymerase I replaces the RNA primers with DNA, and then you have DNA ligase, and what that's going to do is join all of these DNA fragments together, so it's just one continuous strand made from all of these separate smaller fragments. A couple other proteins that you guys should be aware of. One is topoisomerase, and what this does is it relieves strain ahead of the replication forks. One of the things that I think about with this is if like you're ever braiding your hair and you pull it really tight, that your hair is it's gonna tighten up as you're pulling on it. That braid is gonna get really tight. Well, as you're sort of pulling the DNA strands apart, remember it's in a double helix. So as you do that, you're kind of pulling on that twistedness, and so it's tightening it up. Well, if you tighten it up too much, you're not going to be able to separate it. So this topoisomerase actually relieves the strain of those replication forks sort of pulling the DNA apart. And then you've got helicase. The first part kind of looks like helix, and that makes sense because helicase is going to unwind the DNA double helix. And then you've got the single strand binding proteins, and these help hold the DNA strands apart as replication is taking place. Table 16.1 is in your book. I highly recommend you guys take a look at this. It gives you a really good breakdown of all of the enzymes and what they are used for. So here we sort of have like our big picture, right? So we have our parental DNA is the dark blue strand. You can see here that this is the replication fork. This is where the DNA is splitting apart. So this top strand, the original parent strands, is 5' to 3' over here. And so this complementary strand that's being added, this is going to be the 5' side on this part. So you can see we have our RNA primer. And now we have DNA polymerase III adding DNA nucleotides, moving towards the replication fork. So this is our leading strand. This is just continuous adding of DNA nucleotides. Now down here, this parental strand on the bottom is going 3' to 5'. And so what that means is this side, the new strand, is trying to add 3 to 5, which we know that can't happen. And so we are going to have lots more enzymes kind of working here. And so we are going to have primase that's priming this area right here. Once this primer is done, then you'll have DNA polymerase come in and start adding the DNA nucleotides. And that's what's happening at this segment right here. So you can see the primer is complete. DNA polymerase III is adding the DNA nucleotides. It will do so until it bumps into this primer here. All right, then you have, this would be DNA polymerase I, and you can see that it's replacing the RNA with DNA. And then you've got DNA ligase, and you can see here it's working on this split between these two Okazaki fragments, and so it is going to join these two fragments together so that you have just this one long continuous strand of DNA. All right, so the leading strand, this is all pretty seamless. You've got DNA polymerase III just adding bases in the 5 to 3 direction. Down here, you have to have the primase come in and prime. Then you need to have the DNA polymerase III make a short segment. Then you need to have the DNA polymerase I remove the primer, and then you need DNA ligase to join it all together. So this is just kind of just an overview. Again, we have our leading strand and we have lagging strands. Remember that this is happening in 2 directions, so you know, depending on which direction it's moving, the replication fork is moving, that sort of depends on whether you're looking at the lagging strand or the leading strand. But that's sort of the general breakdown of elongation. So your lecture question for today, I want you to practice explaining in your own words the process of elongation, and please make sure that you include all of the appropriate enzymes and what their job is. So this isn't, you know, I'm not asking you guys to you know, solve a problem here. I really just want you to practice walking through these steps of DNA elongation because it's really important, and there's lots of different enzymes, and it gets kind of complicated when you're talking about leading versus lagging strand. So respond on the website, send me a message, and explain in your own words the process of elongation. All right, guys, thank you.